First off, thank you. It's a pleasure and an honor to be here to go through and walk through some of where we are with Homology Medicines. Be aware of our forward-looking statements. Any questions of these, I guide you to our SEC documentation on our investor site of our website. I want to start here with the mission of Homology Medicines. And I think here, with the technology that we have and the tools that we have available, and I think all of us at a meeting like this today, we truly are, all the employees in the company are trying to bring these cures to these patients. That really is what drives everyone to come into work and really work on these technologies in order to bring these forward. And I'll walk you through what we have as a technology platform. Homology Medicines is a fully integrated gene therapy and gene editing company, and we currently have 3 programs in the clinic. One of the things that we really broke it down into 3 components: R&D, manufacturing expertise, as well as rare disease experience. So we have a lot of rare disease experience. We have more than 11 rare disease drugs with about $2 billion in revenue. Coming over to the manufacturing aspect, the company was founded based on the discovery of 15 novel AAV serotypes. And one of the things that we felt that was important right from the get-go is to really understand how well those package manufacture from a process development perspective all the way through to a GMP manufacturing perspective. Taking all of that aligned with our R&D expertise, we've been able to drive these 3 programs into the clinic since the company founded in 2016, as well as 2 additional development candidates. And I'll walk you through how we built that portfolio. Homology right now is also positioned for success. So we're on track to provide updates on our PKU in vivo gene editing program that's currently in phase 1, as well as a Hunter syndrome gene therapy program. We'll give updates on these by the end of the year and where we are with these particular programs. Secondly, we're also very well cash positioned to give us a runway into the 4th quarter of 2024, and that allows us to get through many of these key milestones, not only clinical data on HMI-103, which is our editing program, as well as HMI-203, which is our Hunter syndrome program, but also to move forward our IND-enabling program in a vectorized antibody against anti-C5 for PNH. So all of those programs are well funded to get through that. We also had $130 million in non-dilutive capital from formation of an AAV manufacturing business. This was something as we really focused in developing not only process development but GMP. This was something that we felt we could leverage that expertise for our programs, but then also in forming a collaboration with Oxford Biomedica and do that joint venture. This is something that could be made available to others in the gene therapy and gene editing community. From a partnering opportunity, we just recently nominated a development candidate for MLD that we're actively seeking partners for. And then finally, I talked about the expansion of our technology platform in order to express full IgG molecules out of the liver, and I'll show you a little bit of data on that. The company, again, was founded on these 15 new AAV serotypes, and we went through and we characterized these in non-human primates. We could understand how well do they manufacture, how well do they biodistribute, what tissues do we get into, and can we align those AAVs with a particular disease perspective that we're interested in. In doing so, we were able to set up the company to target genetic diseases using one of 3 platforms. On the left was a traditional gene therapy. You can see there we're using the AAV as a drug delivery vehicle, if you will. We've engineered and worked a lot on the design of the inside of that so that we could utilize a promoter and express a transgene. You can see that on the left. In the middle is an extension of the gene therapy, but instead of expressing a transgene that's missing, we're actually expressing a full-length antibody. And here, the team, again, working on AAV as a delivery approach, engineering the inside of that vector genome so that we could get high levels of antibodies secreted. And then finally on the right, and this is what I think is unique to very few companies, Homology Medicines, I think it's a leader in this area, is on the use of nuclease-free gene editing. And what we do here is we take advantage of the single-stranded genome of AAVs and integrate directly into the genome. And I'll talk to you about our clinical program in PKU that's taken advantage of that particular technology. So with all of that, this is the current pipeline that we have. You can see that we're an adult PKU initially is a phase 1. Our plans for that with the gene editing are to test in adults, identify a safe and efficacious dose, and then rapidly expand into the adolescent and pediatric. And we feel that having the integration into the PAH locus in PKU will allow us to do that so that we can get into a younger patient population where we can continue to have that durability through that rapid cell growth during childhood development. If you continue down into the gene therapy, you can see here we're in phase 1 in our Hunter syndrome program. This is MPS II. That one also, we're in a phase 1 trial in the US as well as in Canada for that particular program. I mentioned the partnering opportunity around another lysosomal storage disorder, metachromatic leukodystrophy. And then we've learned a lot from our lead program in PKU, which didn't integrate, but we went into patients and we were able to see that it was generally well tolerated, as well as seeing evidence of biologic activity at certain doses. So we take all those learnings and we're implementing that into our pediatric adult PKU study in editing. And then finally, the GTX mAb, and I'll walk through a little bit of data on each one of these. For HMI 103, this is our editing program. So this has a unique mechanism of action in which we are able to provide a functional copy of PAH, which is the enzyme that's missing in PKU. And what we did was this, we used a liver-specific promoter. And the reason we did this is when we integrate into the gene, PAH, we wanted to get more expression than we were getting from the endogenous allele. So we put a promoter on there in between the integration. The other aspect that we did is that by going in and knocking out one of those alleles, in a sense, for each of those cells that you do integrate, you're turning that cell into a heterozygote cell. So you're getting more expression of PAH, and you have the heterozygosity of that cell. And then finally, one of the things that we learned early on is that because we're using AAV, and these are single-stranded genomes, each cell that we integrate in has the ability to integrate, but also we form these episomes that are natural to AAV. We wanted to take advantage of that. So by including the promoter, we get kind of a dual approach, expression off the integrated, as well as expression off the episomal allele. And that really led to a significantly more potent molecule. This is a little pictorial of this, how this works. We're using our AAV HSC that has tropism to the liver. We have the homology arms, which are DNA sequences that are about 1,000 base pairs on each side. So it's a total of 2,000 base pairs that are homologous 100% to the intron 1 of the PAH gene. And if you look on the right of that, you can see that it targets that region of the genome, integrates, and then we can get expression of that. If you look to the left part of the nucleus, you can see that those that don't integrate will form an episome. We can also produce a functioning PAH off of that. What's unique about this is that it's really dependent on homologous recombination. So we've done extensive sequencing through this to show that we're not introducing any additional mutations than the cDNA that we're inserting into the gene. And this is really important when we're thinking about turning a gene on. We can't have an introduction or a deletion of a nucleotide that could ultimately take it out of frame. And this was a key component of going after that particular disease. I'm not going to go too much into the data here, but I just wanted to highlight on the left, we can do a single administration of this in the mouse model, and we can basically correct the phenotype for the life of the mouse. And if you look on the right, we were able to show this in a humanized mouse model that we integrate and show expression dose-dependent in human cells, even with an in vivo perspective. We're currently in a phase 1 trial with HMI 103, the Phi edit trial. This is a one-time in vivo nuclease-free. It's a dose escalation trial, again, that's targeting adults with PKU. These are adults with classic form of PKU. The goal is to establish safety, efficacy in that population, then rapidly move to the younger population. Again, we'll get program updates by the end of the year, but this one's rapidly going on. I'm going to switch gears and move to the MPS II program. This is a high unmet need for MPS II. There are several of us at Homology that have been working in the MPS II field for quite a long time, and one of the things we know is that enzyme replacement therapy certainly has brought some benefit to these patients, but what we also know is that there's still significant unmet medical need, even in patients that have been dosed with enzyme replacement therapy for their whole entire lives. So we wanted to utilize our approach to express the IDS enzyme, iduronate 2-sulfatase, that is missing in these patients with MPS II. Because it's missing, they build up an accumulation of what's called glycosaminoglycans in the cells, and that's ultimately toxic to the cells and leads to a variety of phenotypes. What's important is that those subjects that are on ERT have some of the peripheral, but they're still experiencing quite a bit of peripheral disease. So this is a systemic disease that these boys are born with. And even on ERT, they can have chronic joint pain, lung and cardiac conditions, hearing loss, a host of conditions that continually get worse as they age. Also, the enzyme replacement therapy doesn't cross the blood-brain barrier, so there is a neuronopathic form of this disease in about 60% of boys that have Hunter. And so by utilizing an AAV approach where we can do an IV administration that crosses the blood-brain barrier as well as treats the peripheral, we can target all of those with one particular molecule. This is a pictorial of what that looks like. Again, on the left, using the AAV as a drug delivery, we engineer the inside so that we get expression of I2S in any cell that the AAV transduces. More importantly here, those cells that we transduce can also make it and secrete it. So we can also take advantage of neighboring cells that may not have been transduced, but they can also benefit from uptake of the enzyme that's secreted. Here we're moving in toward with trials in adults, and then moving to younger patients once we establish that safety and efficacy. This is a little bit of data here just from a preclinical perspective, highlighting what we're able to show in the mouse model. If you look on the top left, you can see here that we see biochemical correction. So in every tissue that we evaluate, we see a reduction and normalization of these glycosaminoglycans, that substrate that builds up. If you look to the right, by doing so, we correct phenotype in the animal model. So we can look at it with respect to joints as well as skeletal features. And if you look on the bottom left, we also show that we cross the blood-brain barrier and we see these glycosaminoglycans decrease in the CSF. So this was the data that we utilized to support our IND filing at the end of last year. So here again, HMI 204 is another lysosomal storage disorder. This is one that we're actively partnering. This is a little bit behind the HMI 203, which is in the clinic. This is targeting a disease that's caused by mutations in the aryl-sulfatase A gene. Again, here it's a single IV administration in the murine model of disease, shows broad distribution, expression, reduction of those sulfatides that also has led to significant improvements in the vector yield and packaging of this particular molecule. And so we're actively seeking a partner for this program in the lysosomal storage disease space. I'm now going to switch gears a little bit to the third arm of our platform, which is the GTX MAB part of the platform. And in this component, what we were able to do is, again, utilize the AAVs as a delivery. We engineered the vector genome in that so that we could get an expression of a full heavy and light chain IgG. And in this case, it's an IgG against C5, so it's an anti-C5. What you can see on the right, after a single administration with this, in two different mouse models that are important. One's a mouse model that's immunocompromised. The other's also immunocompromised, but it's transgenic for the recycling receptor that all antibodies utilize in the human body. What we wanted to show was that after that single administration, we could see significant levels of this antibody that are well above what we would expect to need therapeutically. It's about tenfold, almost tenfold more than what we would need therapeutically. So we have a very big window of opportunity to deliver this GTX MAB. This is our lead program as we're going forward. We see tremendous opportunity for the use of this for other antibody type programs. Here in the single IV dose in the murine model, we showed again full-length antibodies consistent with those levels, sustained expression, and these IND-enabling studies are underway as we speak, working with the FDA as part of a pre-IND and getting ready to move into our tox studies next year. And then finally, I'm going to talk about the characterization of our platforms. We are a platform company. We have these 15 AAVs, and actually our founder's gone through and shuffled some of these to even identify other properties of these AAVs. And so we can utilize how they biodistribute, understand how we design the vector genome in them, whether it's promoters or microRNA binding sites, as well as a safety profile that we've learned from our clinical experience to date, to then select what will be that next indication going forward. I just want to highlight a small piece of data here that we just recently published, in that one of our AAVs we identified and mapped out that after an IV administration does not target the liver, but we do get significant biodistribution through skeletal muscle as well as the CNS. So this is one where if you don't want to go to the liver, you may be able to dose a little bit higher in order to get those others while staying away from any potential administration in the liver that you would want to avoid. So finally, I'll leave you on the last two slides. I think we lead the field in scientific and clinical innovation. We're the first company to have a gene therapy in PKU in the clinic. We're the first company to have a gene editing program in vivo in the clinic in PKU. We're also the first to have a systemic MPS II gene therapy approach with the potential for crossing the blood-brain barrier and targeting CNS. And as I spoke before, by honing in and investing on the manufacturing front, I think we're one of the first companies to scale to 2,000 liters with the HEC-293, and we established a very unique collaboration with our friends and colleagues at Oxford Biomedica to form Oxford Biomedica Solutions, which we're also joint owners of with that program. And then finally, in the field of AAV-mediated HR non-nuclease editing, again, one of the leaders in the clinic with an in vivo approach for that. So as we go into at the end of 2022, you can expect program updates from our phase 1 dose escalation study in PKU. We'll provide updates on that, as well as our MPS II gene therapy program. For the monoclonal antibody, we'll get feedback from the FDA and progress into our IND-enabling tox studies for that program as we continue to move that to the clinic. And then for MLD, seeking a partner for that lysosomal storage disorder. So with that, I'll end and thank you.